welcome back, everyone. We're almost at the end of the day today. We will now proceed with the last two talks of the day. Our next speaker is Dr. Samarjit Patnaik. He's a group leader at the early translational branch at NCATS. And he will be talking to us today about lead selection and optimization by medicinal chemistry. Welcome, Sam.
Thank you, Sareen. Can you guys hear me properly? See my slides?
Yes. Yes.
Yep, sounds good.
All right. Hopefully this thing on the top will go away once I click. So you guys see the heading, okay?
Now we're on your card.
The title is visible, right?
Yes.
Thank you. Welcome to the last leg for the day. And I hope to capture attention before Shin finishes up the day. So as a medicinal chemist, I often question my place in this forum of assay biologists who are discussing all details of many different types of assays and technologies and how we should be paying close attention to the reproducibility and the robustness of these assays. But as a medicinal chemist, what I have realized is that if I have to go to the candidate of my choice, and this could be a tool molecule, which is used for a proof of concept study, or a drug candidate, or a preclinical candidate, in this process, I have to encounter a number of assays. And it is very important for me to know what these assays can do for me so that I can utilize them for my success or failure. And of course, there are some assays, some activities which I don't want to hit, and there are some assays which I want to utilize to progress my compounds forward. So with that, the way I have designed this talk is I have 3 case studies. The first 2 case studies deal with kinase inhibitors. So with regard to kinase inhibitors, the proof of concept already exists. And if you look at this list below, there are several over 30 plus kinase inhibitors which have been approved by the FDA for indications, mostly cancer and some for inflammation. The problem with kinase inhibitor design is that if you are designing an ATP competitive inhibitor, the compound actually binds to this pocket, which is shown here on the left. That is the ATP binding site. It is called the hinge binding domain, and that is conserved across the 500 known kinases. And that is a problem because if we want a compound to bind there, there's a selectivity issue, especially if it resembles the structure of ATP. However, what you can do is you can actually exploit the region around that pocket to design selectivity into your compound. And what I'm showing you here on the right are 2 classic examples of kinase inhibitors, imatinib and dasatinib. And you can see how the orientation in that pocket is very different from each other. Kinases tightly regulate a lot of different processes which ultimately control cell proliferation, division, differentiation, survival, migration, invasion. Many of these players are either overexpressed or they have mutations associated with them which make them constitutively active, and this leads to this unregulated activity and ultimately cancer. So the first case study I'll be talking about is the first medicinal chemistry project I was involved in my career, where I was in GSK and we were trying to develop inhibitors for insulin growth factor 1 receptor. If you look at this signaling cascade cartoon that I've shown here, the way the signaling works through this receptor, which is a tyrosine kinase, is that IGF-1 or IGF-2, which is the hormone, the insulin growth factor 1 hormone, it binds to the receptor. This initiates a receptor dimerization event where the beta unit is actually the kinase domain, which is inside the cell. It actually phosphorylates the other subunit, the other beta subunit. So this is called autophosphorylation, which leads to conformational changes in that particular protein. And that leads to the recruitment of several different players. Well, a classic example here is the recruitment of the RAS, RAF, MEK, ERK pathway. And ultimately what that does is it feeds into this nucleus, transcription is regulated, and you have the production of different proteins, which leads to the tumorigenesis. And so there was a lot of literature out there, which is shown on the left, which made it a convincing case for us to go after this target. For instance, silencing experiments had indicated very clearly that you can actually stop the progression and the proliferation of cancer cells in vitro as well as in vivo. But one of the main observations in the clinic was that patients who were being treated for breast cancer with Herceptin, which was actually a monoclonal antibody for EGFR, a subset of patients started showing resistance. And this, they found, was being driven by IGF-1R biology. So it made a lot of sense to go after that target. One important factor we had to keep in mind was that this actually bears close resemblance to insulin receptor. So finding compounds which can distinguish between those two proteins will be very difficult. And if we hit insulin receptor, we have to be cognizant of the fact that we can actually interfere with glucose metabolism. So before starting a kinase project, the first thing we have to ask ourselves is, what are the assays available which will help us monitor the activity of inhibitors? And as you saw this morning, especially the talk by Nathan, he showed us a lot of different platforms. So you can have an enzymatic assay which monitors the displacement of an ATP mimetic, which monitors the transfer of a phosphate to an optimized artificial substrate, which can monitor the concentration of the cofactor, ATP or ADP. And commercial platforms exist which help us do that. You should have a cellular assay if you're going after intracellular target. So you could have a cellular assay which monitors the phosphorylation of a particular substrate inside the cell. You can have a target engagement assay. You can have an assay which measures phosphorylation of downstream targets. You can have assays which measure proliferation as well as transcription. So the enzymatic assay which we had at our disposal is described here. So we had the beta subunit, the intracellular domain of the kinase, not the entire dimer. And this had the ability to actually phosphorylate an artificial peptide, which is shown in cartoon here. In the middle, we had a tyrosine residue, which the enzyme was able to phosphorylate. This peptide was charged with a biotin in the end. And then you actually added an antibody, which has a cryptate European complex attached to it. And when you shine light at a particular frequency, it can actually emit light or emission at a particular frequency, which you can monitor. On the other hand, if you add a donor bead, which is conjugated to a streptavidin, it will bind to the biotin, and it can actually capture this FRET signal and emit at a different wavelength. And you can take the ratio between these two emissions and figure out how much of phosphorylated peptide you have. And once you have that, when you start adding the compound, you see a dose-dependent decrease in the signal, which is an indirect measurement of the decrease of phosphorylation of that peptide. So it's a functional assay. And what you will see throughout the talk is the number which is shown here, IC50, which is essentially the concentration at which you see 50% inhibition of the signal or the functional activity. We also had a cellular assay, which measured actually the amount of phosphorylated receptor, but in cells. So the way this was designed, this is Delphia technology. We actually add compounds to the cells and lyse the cells. The plate actually has an antibody which captures the receptor tyrosine kinase, and then we add another antibody on top which captures the phosphorylated receptor. And that antibody actually has a europium cryptate, again, europium cryptate. You saw it again in the morning as well. Then you add some ligands, and this europium is actually leached out. That leads to a light signal. And again, that light signal is proportional to the amount of phosphorylated receptor you have in your assay. And if you add compound, you should see a dose-dependent decrease in that signal, which is a measure of the functional activity in the cells. So we had these two assays at our disposal. And when we started the medicinal chemistry campaign, the hit we had in front of us is shown here, GSK 049A. And what we did is that we had a panel using the same enzyme assay technology I showed. We had a panel of many different kinases available to us. And when we look at 15 kinase panel, in this case, I'm showing you the activity depicted as PIC50, which is the negative log of IC50. So a number of 6 means the inhibition IC50 is 1 micromolar, 9 means 1 nanomolar. what we observed was that we were hitting several other players, Aurora B kinase. We were hitting insulin receptor, which was expected, and other kinases like JNK1, 2, 3, and SYK. So right off the bat, we actually wanted to avoid Aurora B kinase because in other programs we had seen that hitting that kinase led to severe cytotoxicity. And we were also cognizant of the players who were ahead of us in the game. And so that's the medicinal chemists always look at prior art. And so this is not only to give you an idea of where you are in the program and the technology, but it's very important for assay development because these act as positive controls and help you rank order your compounds, how you stack up against these compounds. Every medicinal chemist, has a dream of having a divergent route, which allows you to make a common intermediate, and then you can add different groups in the top, bottom, middle, if you will, and then diversify at a late stage. That allows you to essentially build the structure-activity relationship very quickly, and you can essentially explore different portions of the molecule. So I don't want to showcase everything here, but just wanted to show that we discovered actually some novel chemistry, but we had a route which allowed us to essentially diversify in this R prime area as well as in the bottom area in green here. And this is the compound progression path. So if you remember that cartoon I was showing you with all the players stacking up against you, this is the actual picture. On the top in yellow, I have all the assays which help us measure the activity of the compound, whether it is with respect to the target of interest or other targets, cellular assays all the way till in vivo efficacy and PD experiments in animals. And in the bottom are assays which help you develop the druggability of your compound, which help you measure permeability, exposure, all the way to toxicity. For the interest of time, I will only have time to discuss the ones which have the stars next to them, as I'm describing you the progression of the compounds in this project. So this is the first data table I have. It has a lot of data, but I hope I can walk you through this. So I'm trying to showcase what happens with these assays when you change just one portion of the molecule, R1. The first column actually has the enzymatic activity against our primary target, IGF-1R. Then we have that Delphia cellular assay I described to you. And here we have the enzymatic assay for Aurora B and JNK1. And what I want to quickly draw your attention to is what happens when we change hydrogen to methoxy. And not only do we improve the activity against our primary target, but we completely dial out the Aurora B activity. So that subtle change in that molecule is able to achieve this phenomenon. And this is reproducible. So we're very confident of this SAR, which we had discovered. You can also see that the cellular assay and the enzymatic assay against IGF-1R are not strictly correlating with each other, but there is a general trend which we can trust. On the other hand, the JNK activity still remains in this chemical series. We also discovered that we interchange this water solubilizing bead over here with different groups, which formulated a hypothesis that most likely this was actually situated at the outside of the pocket, probably residing in the solvent front. So before I actually go and describe to you the next SAR points, I just wanted to quickly talk to you about some in vivo pharmacokinetic parameters. And you will see this later in the next talk with Shin. But essentially, most xenobiotics are metabolized by this class of enzymes called P450s. So these have an iron heme moiety, and they have this oxygen attached to them, which have this amazing transformation they can do where they can take the hydrogen in aliphatic as well as aromatic systems and then convert them to hydroxyls, which are then subjugated to further transformations by phase 2 enzymes where you have the attachment of water solubilizing groups and then you essentially excrete them. And for the chemists in the audience, I have some mechanisms so you can see how this is done by this wonderful enzyme. However, that's a problem for medicinal chemists because when you dose a compound via the IV route, what you see initially is a high concentration, and then slowly with time, you see a decay in the concentration in plasma. So you can take parameters like area under the curve or the concentration at the last time point to rank and compare compounds. Similarly, when you do oral dosing, when you have the absorption phase, you see an increase in concentration. You reach Cmax, and then you have a decrease in the concentration as metabolism kicks in. And again, you can take the area under the curve. And if you divide the area under the curve by the dose, you have the dose normalized area under the curve, and you can use that parameter to compare compounds. And you can also take parameters like the Cmax to compare compounds. You can also use clearance, which has an inverse relationship against the area under the curve in the IV dosing experiment. So when we look at these parameters, I'll show you what happens when we do these very small changes to the structure of the molecule. So the first compound, the lead compound here, has R1, R2, R3 as hydrogens. And if you look at these numbers, the potency in the enzymatic and the cellular assay is very good. However, the clearance in a rat PK experiment is not ideal, and the concentration at the last time point as well as the area under the curve is not ideal. However, if you just install a fluorine atom on R1, which is as small as a hydrogen atom, you see a profound effect on the PK parameters. And then if you put a fluorine atom in R2, you see the same effect, not when you put it in R3. And then subsequently, we discovered a compound where we had a methyl group here and the fluorine atom here. We had a 5-fold improvement in the PK properties of this compound. So even if in the MedChem program, even if you have in vivo assays, you can still have meaningful data if it's reproducible, and you can use that to progress your compounds. So this effort actually led to the discovery of this compound 953, which at that time was a tool compound. And what we found, unfortunately, with this compound was that the therapeutic window was very narrow in vivo studies. And so then we went back to the drawing board and started thinking about one more activity which we had kept in mind, and that was the junk activity. And subsequently, we actually designed this compound, which is shown on the top two compounds, where we put this NN-dimethyl glycinamide group at this position X, and we were completely able to dial out the junk activity. However, using this assay, we made a number of compounds which had very subtle changes to this small little side chain hanging out from the molecule. And we discovered that every little chain was very important. So if you remove the carbonyl, you lose the selectivity and you lose potency against your primary target. If you remove the dimethylamine, you increase the distance of the dimethylamine from this carbonyl. And these other changes, we saw a decrease in the selectivity as well as the primary activity. So again, if you have the right assays, you can get this data, and this can be very meaningful to you. As the project was progressing, we decided to expand our kinase panel. And what we discovered was that we had the expected activity against insulin receptor, which bears close homology to insulin growth factor 1 receptor. But we discovered activity against anaplastic lymphoma kinase. And this was a plus because about 4% patients who have non-small cell lung carcinoma have this chromosomal rearrangement, which creates a fusion protein between EML4 and ALK. And it is ALK constitutive kinase activity which drives the cancer in these patients. So that was great. And there was a patient population which we could go after. And so in the end, we are left with a report card, if you will, of our compound, which was our preclinical candidate. And you have activities against the primary target as well as activities against other parameters, which help you realize that this is actually a drug candidate. Now, just to finish the story, the fate of IGF-1R inhibitors in the clinic is still questionable because that modality of treatment has not been approved. On the other hand, about 4 different ALK inhibitors have been approved for non-small cell lung carcinoma as drugs. So that just tells you if you have a suite of assays available to you, you can potentially, with those platforms, discover co-activities which could be actually useful to you as you progress compounds further. So I'll stop there and go to the second case study, where again, I'll be talking about another kinase inhibitor. In this case, this is the CDK2 inhibitor, SU9516. So the way we discovered this inhibitor for Duchenne muscular dystrophy was actually through a phenotypic screen. So Duchenne muscular dystrophy is actually an X-linked progressive muscular degenerative disease, which affects about 1 in 5,000 young men. Due to mutations in the dystrophin gene, which is shown here in yellow, there's a complete loss of this structural protein in the muscles of these kids. And what I'm showing you here is a cartoon drawing of the sarcolemma, which is essentially the plasma membrane of the muscles. And so they have a number of different structural proteins which link the extracellular matrix to the actin and gives that strength to the muscle fiber. And when you have a complete loss of this dystrophin protein, the muscle is extremely weak. And so eventually all muscles in the body are affected. So our collaboration with Dean Birkin in Nevada, we had the idea that what if we upregulate another structural protein, in this case, this integrin, alpha-7 beta-1, we can probably compensate for the loss of dystrophin. So our assay was such that we actually took one of the alleles of alpha-7 beta-1 and replaced it with beta-galactosidase. And so if you throw in the substrate for beta-galactosidase, you can monitor actually the amount of protein in the cells. And when we did the screening with the LOPAC library, which you saw, we discovered this compound, SU9516, which was a CDK2 inhibitor, which had a profound activity. And what I'm showing you here is a picture of actually the myotubes, which are formed by myoblasts, which are the muscle cells when they line up against each other. And the green signal you are seeing here is actually the beta-galactosidase signal, which is associated with the integrin. And so clearly this compound is upregulating integrin. Then we took this compound to muscle cells from humans, patients, and evaluated the compound in an MDX mouse model, and it worked. And so the question was, okay, so it must be CDK2, which is driving the activity of this compound, and we really wanted to check out the mechanism. So in this case, what we decided to ask ourselves was that, okay, so we know that in the myotubes, the compound is working, and that is the cell line where, or the cells where we really are interested in the activity, because it's a muscle cell. So what are the kinases that our compound is binding to? And for that, we utilize this commercially available technology, which is from this company called Kinase. So Kinase has these probes, which look very similar to ATP, as you can see. They have one for ADP as well, but at the second or the third phosphate, instead of having an OH, you actually have an acyl group. And what that allows you to do is when this probe binds to kinases, there are actually conserved lysines on the outside pocket of most kinases. So serine kinases have lysine 1, other kinases have lysine 2. They're in close proximity and actually can bind irreversibly to this probe. So when this chemistry happens, you lose this part of the molecule, but what you have done is you've actually attached an acyl group, which is in turn attached to biotin. And so when you then come with streptavidin beads and you lyse the cell and you add the streptavidin bead, you can capture the biotin with this linker, which is irreversibly tagged to all the different kinases. And so here I'm showing you in cartoon how it happens. So if you imagine this green ball to be this part of the molecule, it binds, and then this is the biotin. And so then after you've pulled down all the kinases, you wash everything away, and then you throw in a protease, and then you cleave and you run an HPLC MSMS experiment, you can get different peptide signatures coming from these different kinases. If you have a small molecule which interferes with the system, actually binds to a target, what you see is the dose-dependent disappearance of that specific peptide which comes from that kinase. So using that technology in muscle myotubes, when we did the experiment, we discovered, as expected, that the compound was hitting CDK2. But let me just quickly show you how the data looks like. I don't want you to read this, but essentially you have this list of peptides from different kinases. And what you're looking at here is the percentage activity, which shows you in red. It shows you how active the compound is and how well it is binding to that particular kinase. And if you hone in on this part of the panel, which is shown here in the bottom, we did the assay in 3 different concentrations, 1 micromolar, 0.5, and 100 nanomolar. We found that we were indeed binding to CDK2. We were also binding to CDK5. However, the binding to CDK2 was acceptable. It was strong, but not as strong as 2 other players or kinases we discovered while we did this. So we were very interested to know what was documented in literature regarding the activity of SU9516, a CDK2 inhibitor, with respect to these 2 different kinases, which we had unearthed, OSR1 and STLK3. And when we looked in the literature, reaction biology, which has this platform technology, which a lot of us use, which utilizes recombinant protein, which monitors the ability of the recombinant protein to transfer a phosphate from a radio-labeled ATP molecule. The gamma phosphate is transferred to an artificial substrate. And they had actually published the profiling data for this compound. And when we looked at that, what I'm showing you here is the percentage inhibition of kinases, a subset of them out of 300. We found that CDK5, as shown before, and CDK2 were indeed inhibited. However, the kinases which we had discovered in the myotubes, in the cellular assay, this compound was actually completely inactive. So this illuminates that if you have different assay technologies, you can have very different results. And so just to conclude, there are several assay formats which are available to do kinase research and medicinal chemistry and screening. But you have to be cognizant of which platform you choose, especially cell-based platforms are probably more meaningful than the other platforms which exist. And so this approach led us to discover new potential targets for Duchenne muscular dystrophy. And I just wanted to bring your attention to another landmark paper here, which was published a while back, where essentially you have a very similar technology where they have these kino beads which are decorated with different promiscuous small molecule inhibitors, and they do the same thing. They take cell lysates or even tissues and incubate and then pull down these different kinases. And if you have a compound which interferes with that, you'll have less of that pull down, and you can essentially figure out which kinases your compound is binding to. And what they found was very similar to what we found in our project. Different clinical compounds have very different activities in cells. And if you're thinking about it, you're probably right, because the expression of different kinases is very different in different proteins. So obviously that will be the case, but this paper actually nicely documents that. So with that, I'll go on to the last case study. So changing gears a little bit, we've talked about kinases. This is actually a hydrolase, again, for a rare disease. This is Gaucher disease, which is actually a rare recessive disorder. It is caused by the deficiency of this enzyme, glucocerebrosidase. So this is actually a hydrolase which catalyzes this enzyme, shown here. You have these substrates called glucocerebroside or glucosylceramides, which is essentially a sugar attached to lipophilic side chain. These actually form integral parts of cell membranes and other lysosomal membranes, but they also play a very important role in actually cell signaling. And what happens in the lysosome, which is the recycle center, this hydrolase actually cleaves this and liberates the fatty acid side chain, which can be then recycled. And as mentioned here, what happens in the disease is that there are mutations which either render the hydrolase catalytically inactive or you have mutations like this one, which is in type 1 disease, which has a visceral manifestation, N370S mutation, which actually renders the enzyme proteolytically unstable. So this enzyme is produced, it is actually catalytically active, but it never makes it to the lysosome. As soon as it's produced by the in the ER in the ribosomes, it is tagged for degradation by the proteasome pathway. And so the goal of our program actually was to find compounds which can actually bind to this hydrolase and prevent it from degrading, or to find compounds which can bind to the enzyme and actually activate the enzyme and prevent the buildup of this substrate in the lysosome, which is the hallmark phenotype of the disease. And then just to briefly mention, there are two other even rarer forms of the disease, type 2 and type 3, which have a very unfortunate neuronopathic manifestations leading to mortality at very early age. So the question is, okay, we want to study this hydrolase. How do we do that? And we want to find compounds which bind to this enzyme. So there is a number of substrates you can pick. You can have fluorogenic substrates, have different fluorophores like 4-MeO, which are dark, but when the enzyme cleaves it, you have fluorescence in the blue region of the spectra. You can have a similar fluorogenic substrate where resorphin is liberated to fluoresce in the red region of the spectra. You can have fluorescence tagged starting material as well as product, and you can follow them if you can separate them. So this is more like the natural substrate, or you can have the natural substrate and you can follow glucose levels. So that's the number of substrates. This is the choice you have. You can have enzyme, which is recombinant enzyme, or in our case, we actually had patient samples available with us. So unfortunately, patients have enlargement of the spleen and liver, and they have to essentially go through a splenectomy, which is removal of the spleen. So our collaborator actually had spleen and liver samples in her freezer, and we were actually struggling to find the N370S mutant enzyme. So we decided to actually take the spleen, grind it up, and use the homogenate as a source of enzyme. So that is another choice you have. And so we did screening with the recombinant enzyme as well as enzyme from the spleen sample. And if you think about it, we rationalized that the splenectomy samples was probably more real to the actual system because it probably has other cofactors which are necessary for the activity of the enzyme, like saposin C. So I just wanted to briefly walk you through what we discovered, and I'm comparing here the screening which we did with recombinant wild-type glucocerebrosidase versus the N370S mutant enzyme derived from the spleen homogenate. One thing was very clear, that we found more actives when we used the recombinant wild-type enzyme, whether it was to discover inhibitors or activators, and very few when we used the spleen homogenate. When we looked at the inhibitors with potent or partial inhibition, we find that the recombinant enzyme actually leads to the discovery of more inhibitors. On the other hand, the spleen enzyme leads to the discovery of more activators, which was very surprising to us. And again, here's a cartoon which shows how the glucocerebrosidase is actually nestled in the membrane of the lysosome, and we think cofactors like saposin C help in the presentation of the red substrate to the enzyme, and that's why they are important and help us in the discovery of finding activators. However, we went back, and with all the hits we had discovered, we decided to do a profiling exercise. And what we did is, I'm showing you just the wild-type glucocerebrosidase enzyme. So we had recombinant enzyme with 3 different substrates, and we had the spleen homogenate enzyme with 2 different substrates. So here I'm showcasing an inhibitor on the top left, which shows inhibition only when you take the spleen homogenate, and it's completely inactive against the recombinant enzyme. This one is the opposite. You see activity only when you take recombinant enzyme, and it's completely inactive when you take the spleen homogenate. This one behaves nicely across all different assays. But since we want to find compounds which actually activate the enzyme, because the disease actually results due to either the enzyme being catalytically ineffective or degraded. So we want to actually find activators. We were very interested in this compound, which was our hit. But here again, you see a gradation of this activity, where the best activity is found when we take the spleen homogenate and this substrate, which liberates the fluorophore in the blue region of the spectrum. So just to showcase, depending on what assay you choose, you can have very different activities with different chemical series. So we actually utilize this assay to drive the chemical progression in the medicinal chemistry program. And we published this a long time back, but this assay actually helped us discover ML198, which became the tool compound for our program. And if you have listened to me, and paid attention to the last slide, you saw that the system we had was quite artificial. So it was very important for us to actually verify the activity of this compound in other orthogonal assays. And for that, we actually took this compound and looked at different cells, fibroblasts, as well as, I'm not mentioning here, actually macrophages, as well as dopaminergic neurons for type 2 and type 3. And what we were able to show is that this compound actually is able to increase the activity of the of the hydrolase, glucocerebrosidase, across different cell types with different genotypes. And we were also able to show that it actually binds to the enzyme, which is shown here in red. So this is high content imaging assays. And it is actually able to increase the concentration of the enzyme in the lysosome, which is where we want the enzyme to go and resume its catalytic function. And we showed that for the N370S mutant. So all these different secondary assays helped us realize that the compound which we had discovered was indeed a compound which binds to the enzyme and actually not only increases the concentration of the enzyme in the lysosome, but also increases the activity of the enzyme in the lysosome. So with that, I'll conclude, and I hope what I have been able to tell you through the 3 different case studies is that assays often reduce the disease biology to settings which should be robust, reproducible, and amenable to testing in high throughput, because they allow us to do medicinal chemistry campaigns. But medicinal chemists have to work very closely with assay biologists as well as informaticians who help them understand the data they're getting. And of course, assays can make or break a medicinal chemistry campaign. And with that, I would like to acknowledge the folks at GSK and as well as NIH who contributed to the case studies. And thank you for your time, and I'll be happy to take some questions.
Thank you very much, Sam. I do not see any questions coming in the chat. So please do type in your questions for Sam now. I do not see any questions coming in, Sam. So thank you very much again.